Okay, well, thank you, everyone. It's an honor to be here. I've been here some time ago and other incarnations, and a lot of you are old friends. Some of you will be new friends. A lot of us work together. So I used to work in pharma for some time and for software companies. At the moment now, I work at Elsevier, who is a large life sciences publishing organization, one of the largest. And one of the things we're proud of, the house at Elsevier published Galileo's book. Got a couple of famous authors. So I'll compress this. Actually, start my timer too. Compress this as a longish talk, so I'll just hit the highlights of it. So to jump right into the excitement and life-changing work that you'll hear about. When I was in pharma, particularly, I worked a lot in patient safety, which involved looking at a lot of animal studies and comparing to human studies to find out these bad things happen to animals. Are they going to happen to humans? Should we stop the program? Or in some cases, or most cases, not stop the program, but start looking very carefully to make sure these things don't happen in humans. Because if you think there's going to be an issue in human, you might not find it if you don't actually test for it. So when you see something in animals, one of the responses would be to increase testing for cardiac issues, liver issues, something like that. So my study here is how well do preclinical studies predict safety outcomes. This is partly, you might even say, controversial topic. It's just been coming out, just came out this week in Science Direct. My co-author, who's not here, is Thomas Steger-Hartmann, who is the head of investigational toxicology at Bayer in Berlin. So why are we interested? Obviously, I mean, just at a high level, we can say there should be some interest in, do these animal studies actually predict what happens to humans? If you read some authors, some especially animal rights activists, you'll say, no, they don't at all, you should do them well. People with more measured approach say, well, actually, they do reasonably well, so we should actually keep doing that. But people are interested in it, one, to lower attrition rates of programs, look at historical, and 3 R's, that's reduce animal testing or repurpose. So what we did was a big data approach, looking at all known data. I think it's a first in several areas, but all known data of animals and humans, and comparing them together to actually do the statistics for each individual type of adverse event and each species separately. There are actually about 20 species that are used in animal studies, but only 5 of them, as you'll see, are the ones really common ones. For each event and species, what's the predictive power for humans that we've observed? So I'll look at some results. Obviously, there are limitations to these kinds of approaches. I might not have time to get into all of them in this talk, but there are limitations. But this is just an analysis, an observational analysis of what's our experience been. I see this in animal, I see this in human, how often do they correlate? One of the reasons for doing this course is the attrition in development. A lot of things fail because of safety, and so if you could predict the safety sooner, you can eliminate this 62% on the slide or reduce that so that you have more projects in. You could try to have more shots on goal, so to speak, that would be successful and not be failing here in early stage safety. And you can see some of the reasons, most common reasons that things fail, or at least are reported to fail. People don't always report exactly why they fail, especially before they get published, but in CNS and musculoskeletal areas. And a lot of things fail actually for things that aren't on this reason. That's my experience of rather simple reasons like rash and nausea. They don't kill you. They're not like necessarily a safety thing, but nobody wants to take a drug that gives rashes and gives you nausea. If you could predict those things, you could have a better handle on the success of your drug. There was the canonical study in the field since 2000 is called the Olson study. It's by a large group of authors, a consortium, pharmaceutical company consortium, that looked at a selected number of drugs, but they only had a relatively small number. And they also did not look at false negatives and true negatives in their analysis. So they looked at basically at sensitivity as the metric and specificity and the positive predictive value and the negative predictive value. And even for the limited set of drugs that they looked at, they found reasonable concordance, we call it. Interesting, in this field, you don't necessarily call it predictive power. We use the term concordance, of how concordant is the animal with human, because prediction is actually a stronger term, you might say. So there is evidence from this study that preclinical species predict human toxicities to a certain extent. The data set was rather small, and the number of different kinds of toxicity was rather small. It was grouped into a relatively small organ class level set of terms. But animals don't predict everything, and there is, of course, the animal rights groups and other people say that perhaps they shouldn't be used. So this is a famous study graph comparing positive predictive value and likelihood ratios, two different metrics of how well animals predict humans for a lot of things, and you can't see any noticeable correlation in that graph. There's one reason for that, actually. Positive predictive value, which has been used in clinic for a long time, is not a very good metric to compare different adverse event ratios. Put this in it because it incorporates the prior probability, the Bayesian prior probability into the answer. So the positive predictive value is also based on how often you ever see it at all, the prevalence. But the likelihood ratio actually separates the actual statistics from the prior probability of having seen it, which makes it a better way to compare two different tests. So how do we resolve this contradiction? Well, more data, that wouldn't hurt. More data, solid statistics, and this is the part where I won't be able to have time to explain everything, but agreement on terminology, because if you're categorizing things together, that's 5 minutes so far.
No, left.
Left, really? Because I started mine, it says I thought we were talking for 6 minutes. Okay.
Well, 15 in total per question.
Okay. So we have, to get this data, this is why this is the most comprehensive. We mined every regulatory filing to the FDA and the EMEA ever made. For the past 70 years, every filing, every animal report, every animal adverse event reported, every human report reported for any animal in the FDA. It turns out there's about 1.6 million adverse events from, 1938 to present. And there's no post-marketing data. Familiar with FAERS and FAERS analysis, this doesn't use that. This is only very highly vetted data from study investigators who actually reporting things directly to the FDA. So everything is very carefully vetted to report whether it was actually caused by the drug or not by actual toxicologists. As you can see, there's a list of some of the top species. And so we looked at, basically, this is basic statistics as used for diagnostic tests. Looks familiar, Jeff, I really got to love this Bayesian type analysis. So does an animal observation, how many drugs do we see an animal observation and a clinical observation for the same adverse events, like QT prolongation, something that everyone obsesses about. The false negative, how many drugs do you don't see an animal or human? There you go. So, but this is, again, the same statistics that the FDA uses to approve a diagnostic test for a disease. So how often is the diagnostic test right, which is the TP? How often does it show that you have a disease? Well, you don't really have it, that's a false positive. And how often does it actually miss the disease? So this is what the FDA, again, would use. So the statistics is very well known and very robust and approved by everybody. So I use this likelihood ratio, and it is a measure of how predictive is it? What is the strength of correlation between the animal and the human? The second part is mapping these to higher levels. All of the reports that appear in regulatory filings are generally at what's called the MEDRA preferred term level, which is relatively low. So it's like QT prolongation or hypertension. However, if you look at these kinds of charts, hypertension or QT prolongation, tachycardia here, can be grouped up into cardiac disorders. So you can answer the question, does this drug appear, any cardiac cardiac adverse event in animal translate to seeing that in human at a high level. So we tried that as well, so you could do things at a higher grouping as well as the lower grouping, the preferred term that was actually reported. The result of the analysis is, although the relationships are highly significant, if you roll up to the high-level organ class term like cardiac, if I see a cardiac issue in an animal, do I see it in human? The predictive power at the generalized level is not that good. I can say about that, it's not very convenient, and it's partly, as the publication has coming out showing, is because when the things that get grouped into cardiac in MedDRA are very diverse. Like it's a lot of things that you would be astonished of actually called cardiac issues get grouped in. So part of it is the vocabulary you're actually using to group things together. So like if you looked at it and you picked out 25 things that you say are the most important cardiac issues and rolled it up, you might get a totally different result. Another interesting issue is that people favor particular animals for particular tests. For many kinds of administration site tests and skin tests, these rabbits. For photosensitivity, I learned while doing this, apparently mouse is exclusively used pretty much for photosensitivity. Dog is pretty much used for cardiac tests, the most often. And that, of course, makes a bias in the data. So that's kind of one of the results of this, and again, discussed more fully in the paper, is that there's an unavoidable bias. The data isn't uniformly distributed. People didn't do every animal study for every kind of adverse event so you can compare it to human. But people favor specific animals because of tradition for specific kinds of study. And there are, of course, interestingly, some things that are observed in animals that are never observed in human, and some things observed in human that were never observed in animals. So you can actually start differentiating between some events that are highly predictive, which I have to summarize this. Most cardiac, the animal studies are very predictive for cardiac, partly because people do a lot of them. There's a lot of data for cardiac studies. And in general, they pretty well predict human events. But there are other ones, less studies, where if you see something in an animal, no one's ever seen it in human. And partly this is used for patient safety, in that if I see this in my animal study, should I panic in my program, or should I say, no, that happens a lot in animals, and you don't have to worry about it. So part of this is getting that actually statistical data so you can differentiate between which adverse events you see in animals should really impact human and which have less impact or less risk implied for human. So like here, for QT prolongation, one of the favorites, the likelihood ratio is 10 times. So that can be interpreted as if you see in any animal, actually, QT prolongation, it increases the risk of seeing it in human by a factor of 10 over not seeing it. At the higher level is ECG. That's the next higher level term, ECG investigations, which includes many other kinds of prolongations and parts of the cardiac waveform besides QT. Still 6.9. And if you go up to the high level group term, cardiac, you'll see 4.3. It's not very predictive. It's not strongly predictive. So that's part of that effect, because it's grouping in lots of, if you use standard MedDRA grouping, it's grouping in lots of other things. One of the things on this is the negative likelihood. That's what you really want, is if I don't see it in an animal, did that predict that's safe for human? That's the statistic you really want. Statistically, the answer is no. There's very little statistical evidence that if I don't see it in an animal, that it means it's safe for human. That's kind of sad, but that's the numbers. But if you do see it for many important ones, if you do see it in an animal, it does predict it's safe for human. So there is value, a lot of value. So this is a cool graph. This is, and it's color coded by organ class. The yellow dots are coming out. The yellow bits are different species. So you can see some tests are much more predictive in multiple species versus one species. And so you can identify tests that, with all the data done so far, you could see you could actually do these ones in the middle in any species, and they're equally predictive. The ones on the outside, they only appear to be predictive because predictive for that particular species as observed. And partly that because for many of the ones on the outside here, these are tests that are done pretty much predominantly on rat, and some of these tests may never have been reported, for example, in mouse or monkey before. So in conclusion, is some animal findings are confirmed as being highly predictive, such as particularly cardiac and some liver functions. The negative predictivity, like the fact that you don't see something, means it's safe in humans, is disturbingly low, but there's again, statistical reason for that as well. And as I said, the predictivity is biased by the frequency of animal use for specific endpoints and the size of data and subset for this. So working actually with our collaborators at Bayer, are working also on things using this for also the CDIS, standard of exchange, and vocabularies, because one of the issues, as we talked about, is the vocabulary, having better vocabulary for animal studies versus human studies. We're working on projects like animals are observed to have things like tail biting or cage biting, which is rarely observed in humans. In certain circumstances. So working on the translatability of those kinds of events. So that was the brief overview. I can give you a link. This will be in Science Direct this week. It'll be coming out, the full exposition. So thank you for your attention, and open to any questions. Yeah.